Today, we are very, very excited to welcome Dr. Melanie Shapiro. She's a JDRF postdoctoral fellow in the lab of Dr. Todd Brusco in the Department of Pathology, Immunology, and Laboratory Medicine at the University of Florida College of Medicine. Her research during her postdoc and PhD is focused on the immunology and genetics of type 1 diabetes. Her graduate work included two major projects. One, identification of serological biomarkers for T1D prediction in at-risk subjects and elucidation of immunological mechanisms of T1D pathogenesis in the non-obese diabetic or NOD mouse. As a postdoc, her studies were focused on the application of precision genetics to understand the heterogeneity of T1D pathogenesis and responsiveness to prevention therapies. To date, her work has resulted in 14 co-authored publications, of which 8 she has led as first author. She has experience obtaining independent funding as evidenced by support during her postdoc by JDRF postdoctoral fellowship and a DRC grant, in addition to an NIDDK F31 award during her graduate studies. Congratulations on all that. That's amazing. And she's going to be speaking about a publication that emanated from her graduate studies of insulin-like growth factor-mediated regulation of T cell expansion and function, entitled Insulin-like growth factor-1 synergizes with IL-2 to induce homeostatic proliferation of regulatory T cells. Welcome, Melanie. And I also will just note that Melanie and her colleague was a winner of the 2021 D Challenge that we hosted. And so that really entailed them taking extra time out of their already busy schedules to sort of dive into pre-existing historical data surrounding T1D and pull out a novel hypothesis, which was judged by a panel of experts to be very valid and interesting in its own right. So congrats on that as well. And thanks for joining us today, Melanie.
Thank you, Monica. That was a really nice introduction. And I had a really good experience with the bioinformatics challenge. This will be a little bit different. There's very minimal bioinformatics in this talk. It's more wet lab work, but that's interesting too. So like Monica said, I'll be speaking about a paper that we recently published in the Journal of Immunology titled Insulin-like growth factor-1 synergizes with IL-2 to induce homeostatic proliferation of regulatory T cells. So I don't want to belabor the background on type 1 diabetes, since everyone here probably is very well versed in the type 1 diabetes field. But we all know that type 1 diabetes is a condition that's characterized by T cell mediated destruction of the insulin producing pancreatic beta cells. And patients require exogenous insulin therapy, either through injections or subcutaneous insulin infusion. And neither of these are a cure. There are, at this point, a few promising immunological prevention therapies. Of course, we know that teplizumab has been recently approved by the FDA, and there are some beta cell replacement therapies in the pipeline. But these have not really been implemented in a widespread manner yet. And so a lot of researchers are really looking at trying to better understand the pathogenesis of type 1 diabetes and come up with some additional prevention therapies as well to potentially complement the existing strategies. All right, so as a brief reminder, there are defects in Treg survival, proliferation, and function in type 1 diabetes, although there are sufficient Treg numbers in individuals who have type 1 diabetes. And low-dose IL-2 has been proposed as a means of selectively rescuing these Treg deficits, since Tregs constitutively express the high-affinity subunit of the IL-2 receptor, which is IL-2 receptor alpha, or CD25. And this therapeutic strategy is really relevant for type 1 diabetes as I'm so sorry, my dog is going crazy. As genome-wide association studies have identified single nucleotide polymorphisms that are associated with multiple hits in the IL-2 pathway, and this includes IL-2 receptor alpha, as well as IL-2 itself, having hits in type 1 diabetes GWAS. And interestingly, before we even sort of came to this idea ourselves, many other very important individuals in the type 1 diabetes immunotherapy space proposed that IL-2 may be more effective as a combinatorial therapy, particularly because there are some issues with high dose IL-2 where there are some off-target effects. And so the idea is that if you combined IL-2 with another therapy, you could use lower doses of IL-2 to selectively target Tregs. And if you had a second agent, maybe that could either benefit beta cells themselves, or you could combine with another immunotherapy. And so what we decided was to propose to use insulin-like growth factor 1 to augment low-dose IL-2 therapy. So insulin-like growth factor 1 is a hormone that's produced primarily by the liver, and it's temporally regulated. What I mean by this is that levels of expression of this hormone peak during puberty. And this is important for type 1 diabetes because this is a time where we know that type 1 diabetes is commonly diagnosed. And like I said, it's mainly produced by the liver, but it can also be produced locally by many tissues, including the pancreas. And much like insulin, IGF-1 is known to promote proliferative signaling, but it targets somewhat different cell types than insulin because insulin receptor and IGF-1 receptor are expressed on different cell subsets. And IGF-1 production is known to be regulated both by growth hormone as well as insulin itself. And so insulin-like growth factor 1 levels have been studied extensively in people who have type 1 diabetes and have been shown to be reduced after diagnosis. And this is not particularly surprising since we know that insulin promotes IGF-1 expression. However, at this point in time had been minimal studies on IGF-1 levels in pre-type 1 diabetes. And so we decided to use our resources to look at this stage of disease development. And this is a publication from a couple of years ago. So I'm just showing some representative plots of longitudinal sampling from 3 different subjects that were from this tidbit trial, which was an observational study that was performed by Novo Nordisk, and they drew individuals who had at least one type 1 diabetes related autoantibody. They drew them monthly for about a year. And a handful of the subjects did go on to develop type 1 diabetes during the study. And I'll show you what one of their insulin-like growth factor levels looks like. But first, we're just looking at a subject with a single type 1 diabetes autoantibody. So this is someone who has begun to potentially develop type 1 diabetes, but has not gotten to the point of developing dysglycemia. And you can see that while there are some variations in their insulin-like growth factor 1 levels over time, it is relatively stable. So then moving on to stage 1 and stage 2. So stage 1 is the development of 2 or more autoantibodies, and then stage 2 is the development of abnormal glucose tolerance. So I'm combining these stages here because we don't actually have glucose tolerance testing on these subjects. So I don't actually know what stage the multiple autoantibody subjects are in. But when we look at this individual who had 4 autoantibodies, their IGF-1 levels decreased over time during the study. And then moving on to clinical diagnosis. So because these individuals were being observed so frequently, they were actually able to be diagnosed quite early on. And so they were diagnosed by an A1C that was greater than 6.4. And that's denoted by this red line here. And here you can see that in this individual who was diagnosed at their second visit, actually, that their insulin-like growth factor 1 levels continued to decrease after diagnosis. So not only are we seeing insulin-like growth factor 1 levels decreasing after diagnosis, but we did see that in individuals who were in this pre-type 1 diabetes stage, that there also were many individuals whose IGF-1 levels were decreasing prior to diagnosis as well. And so, in addition to defects in the IL-2 pathway that I had discussed earlier, there are likely also defects in insulin-like growth factor 1 action during type 1 diabetes development. So back to our discussion of immunology, insulin-like growth factor 1 has been previously suggested to enhance Treg proliferation. And so here we're looking at insulin-like growth factor 1, which was added to sorted human Treg cultures, and you can see that there are increased cell numbers with the IGF-1 treatment. However, these experiments used sorted cells, so it wasn't totally clear from these studies whether IGF-1 preferentially promoted Treg proliferation over proliferation of other immune subsets. And one experiment that sort of tried to answer this selectivity question used conditional knockout of IGF-1 receptor on Tregs. So we're looking at an IGF-1 receptor FOXP3 Cree mouse. And the particular autoimmune model here was experimental autoimmune encephalitis rather than type 1 diabetes. But regardless, they had mice that were treated with the autoantigens that cause development of EAE. And they had mice that were either treated continuously with recombinant human IGF-1 through a mini pump system or vehicle. And what you can see here is that so first we're looking at the control animals that had EAE without any treatment. That's the gray. So you can see that they have this increasing clinical score for EAE. And then we can compare that to the animals that were treated with IGF. So that's the black with the squares. So they had improvement in their EAE symptoms with the IGF-1 treatment. However, when animals had IGF-1 receptor selectively knocked out of Tregs, which is the black circles, this now looks like very similar to the wild type. So we're no longer getting this rescue by IGF-1. So the results of this experiment are essentially suggesting that the protective action of insulin-like growth factor 1 in this model is being imparted by Tregs.
Melanie, we just had I didn't want to stop you in the mid-slide, but we had a question, I think pertaining to maybe the last slide. James Pearson, if insulin promotes IGF-1 production, why would IGF-1 levels decrease when the individual is diagnosed and would be on exogenous insulin?
Yeah, that's a good question. So yes, they are on exogenous insulin. And I believe that exogenous insulin does have some effect on IGF-1 levels themselves. But in general, the literature seems to have stronger associations between IGF-1 and endogenous insulin rather than IGF-1 and exogenous insulin. I don't know the particular reasons for that.
Okay. Yeah, that's probably something to unpack, I guess, if people wanted to like read through the realms of literature. But that's what seems like is the standard. And then with Kiadi Girder from Boston College, it looked like individuals with 4 autoantibodies seem to have high IGF 0 out of 5 month time points compared to 1 autoantibody. And I guess she's just asking for.
Yeah, like in that particular individual that they started off higher and then decreased. So yeah, so for that one individual, that was true. Overall, when I grouped all of the subjects together that had multiple autoantibodies, their baseline was not significantly different.
Okay, great. Thanks for taking those.
Mm-hmm. And then the same person also asked if the individuals with 1 autoantibody and 4 autoantibody are the same. So yes, it's a longitudinal study, but those were separate individuals. So there were some instances of seroconversion within the longitudinal study, but they were not as dramatic as like one autoantibody to four autoantibodies. They were generally loss or gain of a single autoantibody within the 1 year time frame.
Great. Okay, thanks for clearing that up.
No problem. All right, so all this background brings me to my hypothesis, which is that insulin-like growth factor 1 might augment the extent by which IL-2 preferentially induces the expansion of Tregs. And so to test this hypothesis, we wanted to address several goals. One was to define insulin-like growth factor 1 expression on CD4 T cell subsets. The second, to determine if variable IGF-1 receptor and IL-2 receptor expression may impact downstream signaling pathways. And then finally, to use in vitro and in vivo models to assess the impact of the combination treatment on Treg proliferation. So just in the interest of time, I'm not going to show every single figure from the paper. I'm just going to show the most interesting figures, in my opinion. So I'm just going to start off with our first major question in the mouse side of things, which was whether the addition of IGF-1 to IL-2 would promote signaling pathways downstream of IGF-1 receptor or IL-2 receptor. So here we're looking at phospho-S6, which is downstream of the PI3 kinase AKT pathway, which is an IGF-1 receptor signaling pathway. And we saw that the addition of IGF-1 alone had minimal impact on IGF-1 receptor-related PI3 kinase AKT signaling. And IL-2 alone modestly increased PI3 kinase AKT signaling. So PI3 kinase AKT can also be induced by IL-2 receptor signaling as well. But the combination of IGF-1 and IL-2 together significantly upregulated phospho-S6 expression, specifically in Tregs. And despite this synergy, when we looked at phospho-STAT5, which is downstream of the IL-2 receptor, we did not see synergy in this pathway with the combination treatment. So it appears that this is probably downstream of IGF-1 receptor. Could also be downstream of IL-2 receptor because of PI3 kinase AKT signaling being somewhat induced by IL-2 receptor as well, but it hinted towards IGF-1 receptor signaling. So next we asked whether IGF-1 and IL-2 would enhance Treg proliferation in vivo in the NOD mouse. So this was a short NOD study. So pre-diabetic 12-week-old mice, female mice, were either treated with PBS control, IAC, which is IL-2 antibody complex. It's just a means to increase the half-life of IL-2 in the mouse. IGF-1, which was a twice-a-day sub-Q injection, or a combination treatment. And our treatment regimen was IAC for every other day for 7 days, and then IGF-1 was twice a day, every day for 3 weeks. And these timelines were modeled off of previous publications, and treatment regimens were modeled off of previous publications. So what we found was that after that first week of treatment, there was significantly expanded Tregs in the peripheral blood in animals with the combination treatment. And actually, we only saw a trend for an increase in Treg percentage in the IAC or IGF-1 alone conditions. Out to 2 weeks, we started to see contraction of that expanded Treg population in the combination group. And then by 3 weeks, the effect was completely disappeared. So we also stained for Helios in order to distinguish between peripherally induced and thymically derived Tregs. And what we observed was that the expanded Treg compartment in the combination therapy group was likely due to expansion of the Helios positive thymically derived Treg group, as opposed to the FOXP3 positive Helios negative peripherally induced Treg subset. Of course, we also stained for additional immune subsets to check for off-target effects, and we were happy to see that we did not observe any enhancement of off-target effects above IL-2 alone. So here we're looking at CD4 and CD8 T cells. This is just gating on the subsets, and then on the right, we're looking at Ki-67, a measure of proliferation. And then we also, of course, we looked at Tregs. We did see enhancement of proliferation in the Treg compartment. And we did see some proliferation of NK cells. However, in the combination therapy, it was not significantly increased above the IL-2 alone condition. So we next wanted to understand why this synergism was occurring. And so to do this, we measured levels of IL-2 receptor subunits as well as IGF-1 receptor to see if either of these were upregulating expression of any of these receptors. And indeed, we observed that there was increased CD132 or IL-2 receptor gamma expression in the combination treatment. And this was actually specific for Tregs. So here, this was a treatment of bulk splenocytes, and when we gated on naive T conventionals and as well as memory T conventionals, which I'm just not showing here for sake of space, we did not see a significantly increased intensity of IL-2 receptor gamma in either of these populations. So it appeared that this was specific to Tregs. So now moving on to human cells. This is where it started to get a little bit different from the mouse, actually. So we started to look at IGF-1 receptor expression on various immune cell subsets. And what we found was that IGF-1 receptor was higher on naive CD4 T cells than memory CD4 T cells, but that specifically in the naive Treg compartment, that expression of IGF-1 receptor or CD221 was actually the highest. And while IGF-1 receptor expression was associated with age, with younger individuals possessing higher levels of IGF-1 receptor, it was not associated with disease status. So here I'm comparing first-degree relatives to individuals with type 1 diabetes, and there were no significant differences. So next, we wanted to perform some Treg proliferation assays with human cells. And so what we did here was we enriched naive CD4 T cells and stained with a proliferation dye and treated with IL-2 and or IGF-1 for up to 9 days. And what you can observe here is that treatment with IGF-1 or IL-2 alone induced none to very minimal amounts of proliferation of naive conventionals and Tregs, but the combination therapy seemed to induce significant upregulation of Treg proliferation, which is evidenced by this dilution of cell trace dye, and then quantified by division index. And when we look at the FOXP3 and Helios staining here, again, just you can see this expansion. And again, with quantification, there was a significant increase in the percentage of this double positive FOXP3 Helios positive population. So this is one aspect that was consistent between mouse and man, was this expansion of thymically derived FOXP3 Helios double positive Tregs. Okay, so finally, we were interested in testing whether the addition of IGF-1 to IL-2 or IL-7 would augment the transduction of CD4 T cells. So here we're transducing total CD4 T cells with TCR that's recognizing a type 1 diabetes relevant epitope, which is glutamic acid decarboxylase 65 in the context of HLA-DR4. And we use this as just a model TCR for type 1 diabetes, but theoretically, this approach could work for any type of TCR transduction or transduction of any gene of interest. And what we found was that treatment with IL-2 and IGF-1 significantly enhanced the proliferation of transduced Tregs, just like we saw earlier with the non-transduced Tregs. And we also performed similar experiments with IL-7 plus or minus IGF-1, and we again saw that the addition of IGF-1 was beneficial for proliferation here, but this was more effective for promoting the proliferation of conventionals as opposed to Tregs. So despite these increases in proliferation, when we looked at the percentage of cells that were transduced with the specific TCR, we did not observe any significant differences. And so this suggests that the induction of proliferation was generally nonspecific in the sense that it promoted proliferation of transduced and non-transduced cells in the mixture. We were able to confirm that after the transduction protocol and the treatment, that the cells were able to retain naivety, which we think is important for use in downstream assays, particularly if you wanted to study primary activation events. This would be a nice means to do so. And we also noted that the combination treatment with IL-2 and IGF-1 significantly enhanced FOXP3 expression in the naive Treg subset. And this suggests that this treatment might be enforcing Treg suppressive function. We haven't formally tested this, but we think that this is a promising hint at this definitely at least not having a negative effect on Treg function. All right, so to summarize what I've shown, IGF-1 can synergize with IL-2 to promote the proliferation of naive Treg. IGF-1 can also synergize with IL-7 to promote the proliferation of naive CD4 T cells, and in particular, naive conventionals. And mechanistically, we think that this synergy can likely be attributed to increased IL-2 receptor gamma chain expression by IGF-1. And so to conclude, IGF-1 and IL-2 is a novel strategy to promote the proliferation of thymically derived Tregs, both in mouse and man. IGF-1 may sensitize naive CD4 T cells to common gamma chain cytokine signaling. And we believe that IGF-1 plus IL-2 or IL-7 could be exploited for gene transfer or gene editing of primary T cells in order to study primary activation events, or potentially to avoid exhaustion of T cells that are used for adoptive cellular therapies. So I'd like to acknowledge everyone who helped to make this work possible, in particular, Leanna Peters, another postdoc from the Bresco lab, performed a lot of the transduction experiments. Matthew Brown, who is a technician in our group, assisted me with many experiments. The in vivo studies in the non-treatment were performed by Allison Bayer's group back when she was at the University of Miami. She's now with us at University of Florida. Amanda Poskai from the Atkinson Lab group, who was our science writer, as well as our physicians, Dr. Holler and Schatz, who recruit patient samples for us, and our funders. So this much of this work was actually funded by a training grant from my graduate studies, and then kind of extended into my postdoc, um, and turned into this story that you see today.
Yeah,
So
and I just would really like to add that I've added the recent paper, October 1, 2023 from J Immunology that really, fleshes out the story, and is a great paper for those who want to kind of dig deeper and
okay,
spend
great.
time in it. So that's in the chat. Okay, so we have another question. Wondering if you have determined the diabetes incidence in NOD mice treated with low dose IL-2, IGF-1 alone, and in combination.
Yeah, that's the question, right?
That's the
That's
big
everyone's
question.
question. That's the big question.
Yeah.
Yeah, we're applying to grants to get some money to do that.
Next steps.
Uh,
Yeah, absolutely. That's very exciting. And I know that the Brusco lab just has a really great group of people and a lot of power to accomplish these studies. Is this study something you'll be participating in, Melanie?
Um, Probably to some extent. I've like you kind of hinted at, I've moved on a bit to more of the bioinformatics side of things, but I do mentor a lot of grad students and postdocs who do a lot of bench work. So I'll probably be mentoring a student through a type 1 diabetes incidence study. That's the most likely scenario.
Fantastic. Yeah. Now the bioinformatics is quite important at this stage in the game. Don't see any more questions. I guess I just would add that together with Felix Braden's group at the Air Data Commons and iReceptor Portal Access, and several other scientists, including the Brusco Lab and Aaron Michaels Lab, and a number of other labs, including a few in England and Spain, we are putting together a T1D, the first T1D T cell receptor TCR BCR data repo that is standardized and harmonized. And we thank the Brusco Lab for being leaders, and Melanie for your work in getting those TCRs ready and these data, kind of clean and ready to, upload into that data repository. That's been such a great effort on your part and the team's part. So thank you to the lab and to everyone who's participating in that. And so, it'll give the field more data points, basically, we hope, as it grows and evolves.
Yeah,
So, yeah.
absolutely. I hope that lots of people are able to download the data and analyze it themselves and find something interesting.
Yeah, I think we have about almost 100 million, sets in there at this point. So that's really good. Okay. Well, thank you very much. It looks like you've got a couple of accolades in your chat. Excellent job. And we will, great talk. Congrats on the paper. And we can't wait to see what you do next, Melanie. Have a great rest of the day, and thanks for sharing your work.
Thanks everyone for the great questions.